Today we're going to talk about carbon catabolism. Specifically, we'll look at how glucose is broken down to release energy that can be used for building ATP. To help us keep track of all of the inputs and outputs, we're going to create a table and then we'll draw the process above. Okay, now that we have the table finished, let's start drawing. We're going to use this model of glucose to guide us in this process. This model of glucose has 6 carbons represented by the beads, 1, 2, 3, 4, 5, 6. And we're going to break the bonds between these carbon molecules, releasing energy that can be used for ATP. The first step is always going to be glycolysis. And in glycolysis, glucose gets broken down into pyruvic acid. Pyruvic acid is sometimes called pyruvate. I put a little 6C here by glucose to show that glucose has 6 carbons. Pyruvic acid has 3 carbons, and so what happens here is that the glucose actually breaks in half, and if you listen carefully, you might be able to hear the pop as this bond is broken, representing the energy being released. We're left with 2 pyruvic acid molecules that each have 3 carbons. During glycolysis, NADH receives electrons and ATP is generated. So let's fill in the table now so we can keep track. The inputs here were glucose and NAD+, which will be used to receive electrons and form NADH. Then the outputs are pyruvic acid, ATP, and NADH. Okay, so there are 2 pathways now that pyruvic acid can go down. It can either go down this classic pathway towards respiration, which goes through the transition step and the Krebs cycle to the electron transport chain, or pyruvic acid or some derivative can undergo fermentation. So we have a split here. We're going to follow this pathway first, the transition step to respiration. So in the transition step, the pyruvic acid, with 3 carbons, is going to get broken down further, forming acetyl coenzyme A and carbon dioxide. Acetyl coenzyme A has 2 carbons and carbon dioxide has 1. So here we go. Pop, that bond is broken and we're left with acetyl coenzyme A and carbon dioxide. Let's do that to the other pyruvic acid as well. And then typically the carbon dioxide is waste and is released from the cell. Acetyl-CoA has 2 carbons. Carbon dioxide is released. And during this process, NADH is also formed by receiving electrons. All right, let's fill in our table. Okay, now acetyl coenzyme A is going to be the input for the next step, the Krebs cycle. In the Krebs cycle, these 2 molecules of acetyl coenzyme A get broken down all the way to carbon dioxide, releasing that final bit of energy, and that carbon dioxide then is released from the cell. In this process, NADH, FADH2, and ATP are formed. Let's fill those steps in on the table. Okay, I want to pause for a moment and just make note of the fact that I'm not making you memorize the numbers of each of these molecules' inputs and outputs. If you were studying metabolism in detail for your career, then it makes a lot of sense to know exactly how many molecules of each of these is produced from one molecule of glucose. But for our course, I just want you to know the general input and output. I want to share with you a little memory tool that I use for remembering the names of these molecules that are formed as glucose is broken down. So we start with glucose. That's G. It gets split into pyruvic acid. That's P. It gets split into acetyl coenzyme A and carbon dioxide. And then this acetyl coenzyme A also gets split down into carbon dioxide. And so we go from 6 carbons to 3 carbons to 2 carbons to 1 carbon. And this GPAC, I'm just going to write it right here. This also can be used as an acronym for Give Peace a Chance. So I always think of the song, all we are saying is give peace a chance. Glucose, pyruvic acid, acetyl coenzyme A, carbon dioxide. And that way I can trace the molecules from glucose all the way down to carbon dioxide and remember them. Okay, let's move on to the electron transport chain. In the electron transport chain, the electrons that were gathered in these previous steps of glycolysis, the transition step, and the Krebs cycle will be brought to an electron transport system that's embedded in a membrane. And the electron carriers will deliver those electrons as a source of energy for creating proton motive force and generating ATP. So I'm going to underline all of our electron carriers in green so that you can see them, NADH and FADH2. And so these are sort of like little taxis that just drive the electrons to the electron transport chain. NADH and FADH2 both do the same job. They're both little electron taxis. Although, of course, we don't use taxis anymore these days. For the most part, we use Uber and Lyft. And so you could think of NADH as Uber and FADH as Lyft. But instead of driving people, they drive electrons and deliver them to this electron transport chain. We'll get into the details of how the electron transport chain works in a later lecture. But for now, we'll just say that those electrons, the energy that will allow oxygen to be reduced into water. So here's our input, oxygen, and the output, water. And during this process, a lot of ATP gets generated. So now let's go down and fill in our chart. I'm putting the little starbursts around the ATP because there's a very large amount of ATP generated here compared to these early steps. These each generate maybe 2 ATP per glucose, and this is going to generate somewhere in the range of 20 to 30 molecules of ATP, depending on the efficiency. And just to make the ATP stand out, so you can see, this is sort of the point of all of this carbon catabolism. I'm going to circle all the places that ATP was generated. These 2 earlier steps in glycolysis and the Krebs cycle, ATP was generated by what is called substrate level phosphorylation, where it's just a straight reaction of adding the phosphate onto the ATP. And here in the electron transport chain, as part of respiration, ATP is generated through oxidative phosphorylation using proton motive force. And the other thing I want to point out about the electron transport chain here is that I showed this using oxygen. This is for aerobic respiration. And this is a really common way, but there could be some other electron acceptor here that generates a different end product. And so I'll write that in as an option. And just note that this is for anaerobic respiration. All right, let's talk about the alternate pathway now, fermentation. In fermentation, pyruvic acid, instead of going down this pathway towards respiration, go ahead and delineate that as respiration. The other option is to go to fermentation. And so in fermentation, this pyruvic acid, or a derivative of pyruvic acid, will get converted into fermentation products such as acids and or alcohols. There's a large diversity of options of different acids and alcohols that can be produced through fermentation, so I'm not going to write them all. I just want you to know that acids and or alcohols are the fermentation products. This process is going to require NADH, And so this NADH molecule that was generated during glycolysis, instead of going to an electron transport chain, the NADH is going to come here and participate in fermentation. And it will donate electrons, regenerating NAD+. And this is a really important thing to regenerate NAD+, so that there's enough electron acceptor to continue glycolysis. So let's fill in our table down here for fermentation. As you can see, the major inputs are pyruvic acid and NADH, and then outputs would include acids, alcohols, and NAD+. And you notice that fermentation itself didn't generate any ATP. And so the only ATP available to an organism that is growing through fermentation will be this ATP that was produced during glycolysis. And if you remember, there's only 2 ATP produced for every 1 glucose molecule. And so this is a much smaller amount of ATP produced through fermentation than there is if an organism is able to undergo respiration. However, there can be some definite advantages to fermentation. If this is an aerobic organism normally that needs oxygen, if oxygen becomes unavailable, then some organisms can switch to fermentation, which doesn't require oxygen, and this allows them to stay alive. Also, these acids and alcohols that are produced can sometimes inhibit the growth of other organisms and provide the fermenter with a competitive advantage in the environment. Okay, so just a quick recap for carbon catabolism of glucose. Glucose can get broken down through 2 major pathways. One is this respiration pathway, where it goes through the transition step, the Krebs cycle, all the way through the electron transport chain, and this could be done aerobically using oxygen as the final electron acceptor, or this could be an inorganic electron acceptor such as nitrate or sulfate or many others, and this is a way of generating ATP. The alternate pathway here is fermentation, and some organisms have the enzymes that allow pyruvic acid or compounds that are made from pyruvic acid to be converted into acids and alcohols, regenerating NAD+, so that glycolysis can power the cell.